everybody. Reporting to you again from the Glamour City, Hollywood.
You have to choose one of the route, either very fast and very cheap, or very accurate while a little bit expensive. So right now in the market, if you want to get accurate test result, you do PCR test. If you are just get the test result as early as possible, then you use antigen test. And in the COVID market situation, Q Health came up with new molecular testing method, which is called isothermal amplification, which is more accurate than antigen test, but a little bit less than PCR test. And in terms of the price point, it is even more expensive than PCR test. So when the test volume is skyrocketing in COVID-19 pandemic, then they had good chance of the business. But once the situation dwindles, then they lose the market opportunity. That's what just happened. And still, PCR test volume is really big. But again, the small decentralized clinics does not have capabilities in providing PCR test on site. They just send out the sample and takes about a day or three to get test result. So as long as there's a market demand for the PCR test, then there is market opportunity.
If you love listening to this show, please consider giving a rating and a review on Amazon Alexa or wherever you listen. We want to continue bringing you this amazing content, and part of our ability to do that means that we need a big audience. Now, it may not seem like much, but rating and reviewing the show will help more people find us, just like how you found this show. Simply on any podcast platform, search for a show, scroll down to the bottom, and push 5 stars. It's that easy. Thanks for supporting the show. Today, I'm joined by Jinyong Lee, CEO and co-founder of Kryptos Biotechnologies. Welcome to the show today.
Hi, Scott. Very nice to meet you, and I appreciate you to have me today.
So let's talk about PCR, or polymerase chain reaction test. That's usually used to amplify specific DNA sequences. Now, traditionally, when you send samples to lab-based PCR tests, depending on their workload or processing time, it could be several days for the results to return. Is that right?
Yeah, that is correct. So you probably got the, no, please go ahead.
Yeah, well, so then, of course, you got the on-site analyzers that could give results in a few minutes to a few hours, but, I wonder how expensive they are.
You mean the on-site test?
Correct.
Yeah, so currently on-site PCR test is not done yet because of the technical challenge, and that is something what Kryptos is working on.
Okay.
So one of the biggest company who provides the integrated PCR test is called Cepheid, and their system costs somewhere between more than $20,000, and per test price is over $40.
And just to get a sense, for $20,000 or $40,000, how many tests could the different medical professionals actually be able to process?
So in terms of speed, it takes, depending on the different tests, but it takes just around an hour. So the test is usually used in the large hospitals emergency department, not in the small clinics.
Gotcha.
So the many companies wanted to get into this small clinics market, which has a large potential. However, the market requires very fast turnaround time, usually around 20 minutes. So that is a technical challenge.
Gotcha. Okay. So we talked about lab-based, we talked about the costly on-site analyzer, and the fact that it's still not quite meeting that 20-minute requirement. And then you have the rapid PCR test, right? And historically, the issue with the rapid is that there's some serious deficiency when it comes to accuracy and reliability. Is that a fair statement?
Yeah, that is correct.
So can you elaborate why is that the case?
Yeah, so just like other tech industries, there are 3 key values: speed, cost, and accuracy. So if you satisfy all those 3 values, then you will dominate the market. So many companies have tried to come up with a new technology to satisfy those three values, but it's extremely difficult to realize. So some companies just sacrifice the speed, while some companies sacrifice the accuracy. So you may imagine driving a car. So you can drive a car very fast and just pass by the stop line. Or you may drive slowly and stop in front of the stop line. But it is really difficult to drive very fast and stop in front of the line. But you can do it with like a supercar like a Ferrari or a Lamborghini, but they are very expensive. Just like that, the PCR technology is really difficult to do fast PCR cycling while provide accurate control to get the best test result. You may do that, but if the cost of the part is really expensive, it is difficult to make a cost-effective product to be deployed in the point-of-care testing market. Those are the challenges.
Mm-hmm. And just to put it in some context, historical context, so that the audience understands the impact of this, is that when we think about infectious disease diagnostics, and let's take the example, more recent example, of COVID pandemic, where accuracy and reliability in terms of diagnosis it's really a matter of life or death. And how quickly you're able to stop the spread of disease is incredibly important, as we saw with COVID. So with that, what exactly is Kryptos Biotechnologies, and how is your company enabling highly accurate sample to results in under 25 minutes for any infectious diseases, while still making sure that it still has that affordability and can be, in fact, relatively easily placed into smaller clinics?
Yeah. So I usually use this analogy for our product. We make a Nespresso coffee machine for diagnostics. So we have a small box of system, and we have a cartridge to accommodate the patient sample. So once you put the sample into the cartridge and put the cartridge into the machine and run, then you get the test result in about 20 minutes. So in the molecular diagnostics, like a PCR test, there are 3 steps. The first step is the called sample preparation, which extract, purify, and concentrate the target DNA from the patient sample. And then you do PCR biochemical reaction to amplify the number of target DNA. And then lastly, you do the detection to find whether the target DNA is present, is in the sample or not, to determine whether you are infected or not. So in our small box of the system, we have all those 3 steps, and that is the gold standard, which is used in the large laboratories.
Now, there are PCR tests that's able to do multi-diagnosis. Is yours able to do that, or is it focused on very specific one disease at a time?
That's a great question. Yes, our system is capable of running multiplex, which is called multiplex. So our system can detect up to like 6 different targets at once. And if we accommodate a different approach, then we can increase the number of targets up to 15, theoretically. But the biggest question here is whether the market needs those type of large level of tests or not. So in the big hospital, they may be, but in small clinics, usually they require just 4 or 6 targets.
Right. And again, just to kind of layer in a little bit of context, is that, let's say, going back to the COVID example, you would actually have patients that would have COVID, but also they would have, let's say, some sort of a cold, or even lung, some sort of inflamed lung, or something else like an asthma, for instance. So it helps to isolate one or several things that could be affecting somebody's health at that moment, correct?
That is correct. And that is the reason why Kryptos is developing our first target product, which contains flu A, flu B, and COVID-19 and RSV. Those are 4 most prevalent respiratory diseases.
Yeah, makes perfect sense. Now, let's transition to talk a little bit about the technology. My understanding is that the core IP was licensed from UC Berkeley, which again is around this photothermal heating that generates heat, controls the temperature utilizing optical thin film to then efficiently convert that light to heat. How is that process able to leverage the low heat capacity of the optical thin film to then support that ultra-fast PCR thermal cycling that you're talking about?
Yeah, you know exactly what we have. So my technical co-founder, Dr. Son, invented this technology while he was for UC Berkeley. So this technology is called porous thermal heating, which uses light as a heat source and use optical thin film that efficiently converts light to heat and control the temperature. So you can just imagine like in a sunny day, if you go outside, you feel hot because the sunlight converts to heat. And if you wear black shirts, then you feel hotter, which means depending on the observing material, the light to heat conversion efficiency is different. So we either use high light output power or use very efficient material that converts light to heat. That is the option we have. But if you have high light output power, then you have to spend more money to get the strong source of the light, which is laser. So we just use LED light just for the lighting, but use very efficient material that converts light to heat that generates over 100 degrees Celsius. And usually the PCR reaction requires heat up to somewhere around 95 degrees Celsius. So by just combining LED light and optical thin film, we convert the LED light into a heat that is enough to realize PCR reaction.
And again, the reason this is technically a critical milestone is that the traditional PCR tests are utilizing much higher power components. And of course, as a result, those components are going to be much more costly as well as mechanically prone to fail and then larger as well. So talk to us about how this reduces overall cost structure, but also miniaturizes the overall unit.
That's a great question. So in the traditional and conventional approach, usually the company uses the Peltier device or thin film heater, which may provide enough heat, but as you mentioned, it spends a large amount of energy. And also, essentially, all those conventional approaches have to contact between the heat source and the consumable to transfer the heat, right? But our technology uses light, so we do not contact from the light source to the PCR chamber. So with this approach, we can realize very very different types of design, and also it can reduce the cost of the system inside because we just need a LED light and PCR chip is deposited with optical thin film, while the conventional system have the heater and other heat block on top of it, which has high heat transfer efficiency, and then it has a consumable, and then it has multiple systems to not only provide heat, but also extract heat because the PCR reaction have to go up to 95 degrees Celsius and again about 65 degrees Celsius, about 40 times of the cycles. So we have both energy sources. However, our system only has an LED light and small pan because the optical thin film has just like 20 nanometer of the thickness, which has very low heat capacity. So once you turn on the light, it generates heat efficiently. And if you turn off the light, the heat inside the PCR reaction chamber just dissipated through the optical thin film. So it is really fast and efficient structure.
Yeah, so I'm thinking about the traditional PCR components and how this notion of thermodynamics that expands and then contracts and then goes through multiple cycles throughout these diagnostic tests, how that's going to really wear down the number of cycles that it can actually do before it needs maintenance or replacement. So it's not only the initial cost of that 20, 40, or even $100,000, but the maintenance and the replacement costs is going to be pretty high as well, right?
That is correct. That's a great point. And another beauty of using photonic PCR, the porous thermal heating technology here, is that the consumable has higher not only efficiency, but also it has a very low failure rate because usually the heat source covers the entire area of the PCR reaction, then you provide heat stress to the bonding area around the consumable, which makes the failure of the consumable making leakage or something like that. But in our technology, we only provide heat where the optical thin film is deposited. So we do not provide any heat stress to the bonded area. So it significantly reduces the failure of the cartridge, which eventually reduces cost of the users.
Yeah, absolutely. Absolutely. Because, we've heard too many cases where certain PCR tests are really, in some cases, a false reading. And that could, of course, have huge liabilities as well. Now, let's talk about in 2019, I think your company licensed out to LG Chem. Can you talk a little bit about that and what that means?
Yes. So we raised our seed funding from LG Chemical, and they have life science business department inside the LG Chemical. And they wanted to have new technology that increases their strengths in the molecular diagnostics business. So they were very interested in our technology because we have, as mentioned, we have technology very good at realizing fast PCR while having a high level of design freedom because they want to make a very unconventional system. So we licensed out portion of our intellectual property to LG Chemical late in 2019. And they spent about 4 years of the development, but for some reason, they were not very successful in the development and just they returned our license back to Kryptos.
Gotcha. Understood. Now, because you guys have been in business for some time and certainly predating the COVID pandemic, I wonder what type of PCR tests were you able to support during that kind of 2019, 2020, 2021 time period?
I'm sorry, Scott, I didn't hear you well.
Yeah, so the question is, were you able to actually support PCR testing during COVID pandemic?
No, we were not able to do that because when we started this company in 2017, we just focused on developing our technology better. And in 2019, we actually wanted to develop a very unique system for oncology tests or single cell analysis. But in 2020, when COVID happened, we pivoted from our business from oncology tests to the infectious disease testing. So we just did not have enough time to develop a product to launch to market.
Gotcha. And then, of course, there's the FDA process as well, including even if you use emergency pathway, it's still going to take a little bit of time, which we'll come back to in a second. But let's go back to the oncology side, the cancer, which is very interesting. And again, curious to know, in that case, you mentioned single cell multi-omic system, but is there a value in being able to try and detect multiple different types of cancer, kind of multiplexing from that point of view? Again, we're just trying to understand, how it's doing the multidimensional analytics.
That's a great question. So single cell multi-omics doesn't necessarily mean that you will detect multiple cancers at once. Rather, it helps the users to identify certain type of cancer very precisely. So if you just look at the DNA side, you may not have a full picture. Or if you only look at the protein side, you may not have a full picture of the status of the cancer, right? So that is a reason why we check both in genomics and proteomics, which is called multi-omics. And the reason why you want to test in the single cell level is that each cell may have different, like, for example, driver mutations or different status of the cancer progression. So you want to have very minute level of insight on the cancer tumor or micro cancer environment. That is a reason why people want to do single cell multi-omics testing.
Now, tell me about the current limitations of the biopsy and the testing for cancer cells and how your technology will actually improve that.
Yeah. So in current testing, usually they test in a bulk sample. So for example, if you biopsy certain tissue, then it contains like tons of cells inside, right?
Mhm.
And then you just dissociate the cell and test the DNA. Then you only have the bulk insights of the full of the sample. But again, as I mentioned, each cell may have different driver mutations and each cell may have different progresses of the cancer. So you want to have you want to have very low level insights in a single cell level. In this way, you may identify most efficient treatment for the patient. So currently, some companies provide single cell testing, of course. However, they usually use next generation sequencing to analyze the single cell level information. In this approach, you will have like tons of data, which is good, but it costs a lot and it takes time. But once you identify what is the meaningful target, like a biomarker, then you may want to have more efficient and low cost system or approach to follow up the patient progression, right? And that is the area where PCR has to be used. However, in the single cell analysis industry, there are no PCR system that supports single cell level testing. And that is where we want to get.
Okay, so clearly there's a market gap, and just to make sure I understand, so traditionally there's that next gen gene sequencing, right? That's
Mhm.
able to give incredible amount of data sets, but again, it's expensive, it's comprehensive, and again, it's looking at everything. But to your point, where the real play is the follow up and follow on as they're going through,
Mhm.
let's say, certain types of therapies or even treatments that you can actually have very specific biomarkers and see exactly what's happening with that particular cancer cell type versus having to go through that rerun again and again in a very cost effective way. Is that correct?
Yeah, that's the point. So in the
Okay.
beginning of the testing or diagnosis, you may want to run NGS to get large amount of data. But once the patient get treatment, then you have to monitor the patient and follow on with the person for like next 5 or 10 years, right? For that purpose, you may want to have low cost and fast and easy to use system. That is where PCR plays.
And as I'm listening, that's got to sound fairly attractive for healthcare, let's say insurance, right? For those that are having
Correct.
to pay for that. And then of
Mm-hmm.
course the patient themselves are going to have some copay, or in some cases the insurance is even going to cover some of these things. So it has a huge downstream implication in terms of cost minimization of not just a point in time, but the continuity over the life of that particular therapy, let's say.
That's correct. Yeah, that's exactly why we wanted to, yeah, do the epigenetics.
Okay. Now, how difficult is it to tune again, we haven't talked about the algorithms, but tune it for specific biomarkers. So in other words, is it very dramatically different for, let's say, breast cancer cells versus, let's say, pancreatic cancer or something else, for instance? And just trying to get into a little bit more again, I'm not sure if you guys are using machine learning, but if talk a little bit about the algorithm and how you guys actually tune for these specific biomarkers.
Yeah. So finding certain biomarkers or doing the diagnosis in the beginning of the treatment is more suitable for the NGS, as I mentioned, because you may not know what is the driver mutation, right? So for that purpose, you have to go through all the different possibilities at once. That is the area for NGS. And once you identify several uh, like a meaningful biomarkers to follow on, then, for example, Kryptos developed a specific and customized assay for the patient to do the follow-on test again and again and again. So in our view, once you find the meaningful biomarker in the NGS step, then we just translate it into another product for the follow-on. So it does not require any like a very difficult or unique algorithm to do that. It's just like a simple biology.
Gotcha. Understood. Makes perfect sense.
Yeah. However, I want to give a little bit of thought on here. So in the multi-omics, you may identify very unique interaction of unique set of insights when comparing the protein status and the genomic status. Then depending on the status, you may identify another insight. For example, oh, this person may have a recurrence, or oh, this patient may have another mutations that will happen in next 1 or 2 years. In that purpose, you have to run machine learning to analyze whether the patient have a certain level of risk in the recurrence or some type of new cancer happens.
Yeah. So in addition to helping with current therapies, there's this value of predictability as well, which is incredibly important because, again, I think one of the biggest fears with cancer patients is that they may have gone into remission or they think that they beat it, but there's always that nagging fear of is it going to come back or other types of issues that's going to affect them. So this is hugely important. Let's talk about monetization. How are you guys pricing it? What is the business model? Who are the ones that's actually paying for it or co-paying it?
Okay, so going back to our current business, which is the point of care testing market, our business strategy is this model is the razor and razor blade model. Just imagine Nespresso coffee machine. So you provide the system to the small clinics, and when they run the test, then you get charged for each consumable they use. So we haven't confirmed yet, but our system will be provide for example, for free, and then we just make a recurring revenue by selling the cartridges. And the price of the cartridge is largely driven by the reimbursement because it's a clinical industry. So it depend. So depending on the type of diseases, the reimbursement level is different. But the core here is that we want to ensure the users, which means small clinics, make profit while using our product. So for example, for COVID-19 test, they will get about $51 per test. So if we provide, for example, $20 or $30 per cartridge, then they will make about $20 of a profit when they test one test.
Gotcha. Understood. So of course, part of the option is going to be the FDA approval. So can you talk about the different FDA approval process in place? Because I'm sure the infectious disease diagnostics is going to be different from the oncology side, right?
That's correct. So for infectious disease testing, and usually it is called in vitro diagnostics. So it requires just one clinical test. However, if you want to use this system in the point of care setting, your system should satisfy another accreditation, which is called CLIA waiver. So if you want to get the CLIA waiver, the system is really easy to use, even untrained user can use. So not only having a good level of accuracy and performance, you also want to ensure the usability. So just unlike other infectious disease testing or oncology tests, our point of care testing has to have those two aspects, CLIA waiver and 510(k) approval from FDA.
And from a timeline, where are you guys in terms of getting those approvals?
Yeah, so as mentioned, our first target product is respiratory combo test, including flu A, flu B, and RSV and COVID-19. And we are shooting for running clinical study in late 2025, where flu season starts. And then it takes about 4 to 6 months to complete the clinical study. And then it takes about 6 months to get FDA approval. So we plan to receive approval by late 2026 and start commercialization at that moment.
Now, while this process is happening, again, unfortunately, it's just the nature of being in this line of work. What kind of either usability or preliminary kind of feedback are you getting from the marketplace, from some of these smaller clinics, making sure that when things are approved, that you're going to have a captive or a excited user base, customer base that's ready to adopt?
So as mentioned, users want a product with best turnaround time and accuracy and low cost. That's very simple, and again, it's extremely difficult to realize. And there's no product that satisfies those three values yet. So once we get approval from FDA, we want to get into the small clinics like a decentralized clinic, and our first target subsegment is urgent care clinics. So our core approach to them is to provide a product that will enable them to provide better patient service while making more profit.
Okay, gotcha. And again, it's one thing to say that on paper it looks attractive, but are you seeing and hearing that these clinics are ready to adopt when things get approved?
Yeah. So until now, we have collaborated with Stanford School of Medicine to check the usability and performance and potential feedback from their clinicians. And yeah, it went very well because the performance is on par or better than high throughput system, which is used in the large laboratories, while providing test result in about just like 25 minutes. So yeah. So they have, of course, they provided several feedbacks like usability, including like graphing user interface, something like that. But they are just minor change in the workflow. But in terms of performance, turnaround time, size, and everything, the feedback is very favorable, and we are very confident that our product has a competitive advantage in the market.
Okay, fair enough. My final question and kind of comment is that, again, recognizing that it is not a one-for-one comparison, but in the general space, Q Health, which went public in 2021 at some $2.3 billion valuation, recently had to pretty much completely shut down and lay off everybody. And there are others also in the PCR test market that has struggled post-COVID lockdown. And I wonder, do you feel like the space that you're in is still going to be successful and that the market attraction and adoption and the, effectively the sales spike up is going to be there when things are finally approved?
I believe so, and that is the critical question I usually receive from investors or even new employee interviews. So for Q Health's case, I really feel sorry about their failure. But in my point of view in the market, you have to choose one of the route, either very fast and very cheap, or very accurate while a little bit expensive. So right now in the market, if you want to get accurate test result, you do PCR test. If you are just get the test result as early as possible, then you use antigen test. And in the COVID market situation, Q Health came up with new molecular testing method, which is called isothermal amplification, which is more accurate than antigen test, but a little bit less than PCR test. And in terms of the price point, it is even more expensive than PCR test.
Mm.
So when the test volume is skyrocketing in COVID-19 pandemic, then they had good chance of the business. But once the situation dwindles, then they lose the market opportunity. That's what just happened. And still, PCR test volume is really big. But again, the small decentralized clinics does not have capabilities in providing PCR test on site. They just send out the sample and takes about a day or three to get test result. So as long as there's a market demand for the PCR test, then there is market opportunity. The core and key question here is whether you have a technology to deliver it, actually really deliver.
Terrific. So on that note, I have been joined by Jinyong Lee, CEO and co-founder of Kryptos Biotechnologies. Thanks for joining today.
Thanks, Scott.
If you've enjoyed this episode, take a moment to rate our show on any podcast platform that you listen to. Scroll down to the bottom and push 5 stars. It's that easy. And as always, thanks for listening.